You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Jamie Foster. She's an associate professor at University of Florida. We're going to be talking about the interactions between microbial communities and their surrounding environments, and finding out what's going on. You know, what's the microbial chatter and how are they interacting with the environment? What are they eating? What are they excreting? You know, what are these guys doing? Is my understanding. What is the understanding? But Jamie, thanks for coming.
Yeah, thanks for having me, Richard. It's great to be here.
So, I mean, there's lots of microbes, there's lots of environments. What's your focus? Which microbes, which environments are you looking at?
Actually, my focus is I cast a wide net, and the reason is there's so many interesting things to look at, and a lot of times, believe it or not, things dovetail in. I mean, I work on microbes that live in squid and microbes that form rocks out there in the environment, and yet there's a lot of similarities and parallels between them, and it's really a fun occupation, I should say, and microbes are just so important for our everyday lives. I mean, almost everything we eat, everything we interact with, is the product of microbes. One of your previous guests was talking about breathing and hypoxia, and every breath of oxygen that you're taking into your body is actually because of microbes and their ability to produce a lot of the materials and molecules that we need to survive.
So have you said you studied microbes inside of squid. What about inside of people or other animals?
I personally haven't studied humans, and the reason I study the microbes inside of a squid is because it's a lot simpler to work with. And the human microbiome is a very dynamic, it's really such an interesting ecosystem. All the microbes that interface with our body, we call that the microbiome, and we're learning so much about how important the microbiome is for human health. Things like diabetes, obesity, some of our behaviors can all be, it's digestive disorders, all can be attributed to disruptions in the healthy microbiome of our bodies. And there's thousands of different species of microbes interfacing with our bodies, but yet it's really hard to understand what one bacterium A, B, and C is doing. And so to simplify that and work on model systems like the squid system, it gives us an opportunity to really tease apart what one microbe is doing to one host. And that's what's so special about the squid symbiosis, is that it enables a very simplified model system where we can ask more discrete questions about how the microbe is actually talking and communicating with the animal host. And then we can use that insight to make parallels with the human body.
Yeah, so a question I don't have much clarity on is, are you seeing a lot of interaction and cell-to-cell signaling between microbes of the squid and the squid's somatic cells?
The somatic, you mean the reproductive tissues?
No, just any of the squid itself.
Oh yeah, actually
the bacteria just like living there and eating stuff off the squid, or are they talking to the squid cells back and
They're
forth?
actually talking, they're communicating, they're sending molecules back and forth between the tissues. If we counted all the cells in our body, we're actually mostly microbes. 90% of the cells in our bodies are actually microbial. Now, by cell size, the human cells, the eukaryotic cells are bigger, but in terms of sheer number, we're mostly microbes in terms of quantity. So with the squid, just like with our bodies, every surface is interacting. The squid are literally bathed in different types of microbes as they're swimming through the water. But what's interesting is not every microbe colonizes the tissues. They form symbiotic or special relationships they've done this over time, and that's what we call it as a mutualistic symbiosis, where the bacterium is helping the host or the animal do a specific function. And it's the same with humans too. We don't interact with every bacterium, but we've over time evolved special relationships with certain microbes. And for the squid, they've evolved a relationship with a glow-in-the-dark bacterium in order to have the ability to hide from some of their predators. So that bacterium has given them kind of a special power, so to speak, to an invisibility cloak, if you might say, in order to hide from predators. And in return, the bacteria get food. They get amino acids and certain nutrients from the squid in order to survive and live. So that's that give and take in the case of the squid. And similar relationships, we're not glow in the dark, but we have similar relationships with microbes where we get benefits from having their presence in our bodies.
It's weird when a squid encounters a microbe that's not inside of it or a part of its normal functioning. Who's doing the evaluating and the vetting? Is it by committee with the squid cells, the microbe cells, to say this new creature is friend or foe?
Yeah, that is a very important question. Where is the balance between health and disease? Because a lot of the communication signals, we call them microbe-associated pattern molecules, but a lot of the molecules that bacteria and animals use to talk to each other are the same whether it's a pathogen or a beneficial microbe. So the trick here is to really understand where is the balance between health and disease and what tips it in one direction or another? Why does one bacterium become a pathogen and another one become a mutualistic bacteria, like a beneficial microbe? So that's been a conundrum that we've all been trying to understand, this balance of health and disease. And in the case of the squid, the decision-making process, so to speak, is that the bacteria are kind of culled. They're kind of pulled out of the environment and enriched. And what the squid does is as soon as it's born, so the squid is born azenic, meaning it has no microbes in its body or associated with it. So it has to go out and acquire those microbes every new generation that a baby squid is born into. And that's the same with humans too. And what happens is the squid, as soon as it senses presence of microbes in the environment, that activates its immune system. And animals invertebrates have, all animals have an immune system, and some animals have what's called an innate immune system, and they can respond to bacteria in a very generalized way. And so the squid's innate immune system is sensing these microbes and activating some of its responses. And what happens is it starts to produce. It has a special organ where the bacteria live inside. It's called the light organ. And the bacteria start to accumulate. They get enriched because of some of the signals that the host is giving. And then some other microbes might get trapped up into that enrichment process, but they can't survive. The Vibrio fischeri has evolved mechanisms to deal with some of the immune responses or the stress responses that the squid puts on the bacteria. And so through a selection process, the squid kind of selects for this one type of microbe that it knows will luminesce and fluoresce inside its body, giving it this anti-predator behavior, or called counter-illumination. So it's a multi-step process.
How would it know that? How would the squid cells know
Well, they don't know
Oh,
initially.
this bacteria is a good one.
Well, so there's this, it's kind of this multi-layered password protected situation where you have the squid is first the squid is just sending an all points bulletin to say, come on bacteria, come into the vicinity of my little special organ where you can live and get all the amino acids you want for life. So all these different types of microbes come in, and then there's like another signal of a sugar that the bacteria will feed off of. And so, however, in order to travel into the light organ, it's kind of the gauntlet of death here, that there are many different hazards that the bacteria have to withstand. So if you are righteous enough, I guess, for that bacterium, and you can survive the extreme oxidative stress and all the different conditions that the squid is throwing at that microbe, then you are deemed worthy and you can enter the little light organ and survive. So it's the bacteria are then sending back and forth, it's altering its behavior in order to withstand what the squid is throwing at. So the squid kind of lures in lots of bacteria, but it creates a series of physical challenges that the bacteria have to overcome in order to make it past. We literally call them these ducts that travel into these little spaces so that the squid can get populated by this one type of microbe that can survive these challenges. So
it's always the same microbe that makes it to the light organ, right?
Right. It's only one specific type. And if you make mutations in certain genes of this bacterium, you can knock out its ability to withstand. So we can then tease apart what genes, what protein products, what molecules are essential for that bacterium in order to colonize. If it can't handle, for example, that traveling down the duct and into the spaces with it deep within the light organ is riddled with really high levels of oxygen stress molecules. And if the bacteria can't upregulate or make proteins to deal with those kinds of oxidative stress molecules, they die. They just can't survive. So there's lots of give and take between the bacterium and the host in order to establish this relationship. And it's the same with humans too. Go ahead.
It seems to say that there's foreknowledge. There's already an adaptive response that this particular strain of bacteria has made. So time and time again, when new squid come, that it would be able to get through to them.
Well, I mean, there's obviously selection pressure. I mean, if so, mutations in bacteria happen a lot. I mean, in animals, it happens a little bit less frequently, but bacteria are constantly making mistakes, and sometimes those mistakes turns out to be something beneficial. But for the most part, if the bacterium makes a mistake and all of a sudden can't upregulate a certain enzyme to deal with the oxygen stress, it's out, it's gone. So there becomes this constant pressure to always produce these kinds of molecules. And I mean, that's evolution. I mean, it's some sort of variation in the population that some microbes have this gene, and not all Vibrio fischeri are the same. I mean, there's some strains of Vibrio fischeri that can't do this because they haven't undergone these selection pressures. And now another thing that helps this process, maybe will help explain this, is that every morning, so the light organ fills up with Vibrio fischeri and they grow and they start to luminesce. But every morning the squid spits out, it vents all 95% of the microbes it's been harboring all night back into the environment. So that's also adding a selection pressure to cultivate essentially the microbes you want to have come back for the next generation of microbes, for the next generation of squid, excuse me. So it is preferentially seeding the environment around itself in order to for its babies to acquire that special Vibrio fischeri that can glow in the dark and provide the squid with this anti-predator behavior. So this venting process,
so that means that it's ejecting the bacteria that have adaptively responded to the squid's environment that were successful. It's ejecting them, so essentially a pre-informed, when baby squid comes, they're the most likely to make it through because they've already adapted and they know the
Right,
environment, what's ahead.
exactly. It's a massive selection process. Now, the bacteria are just doing whatever it takes to survive. They don't necessarily care what they're doing in some respects, but they're just, okay, this, when I have this gene, when I keep, so there's a certain selection pressure in order to keep becoming luminescent. There's a selection pressure in order to have a certain complement of genes. And if they lose that, then they lose that ability to form that relationship. So there's a lot of, there might be a lot of variation within the Vibrio fischeri population, but that selection pressure of constantly being reseeded and being able to only pick those microbes that can survive this gauntlet of doom here in order to get into the light organ, that's what evolution is all about. It's just adding selection pressure to select for certain different mutations or certain different characteristics.
Well, how about trying the experiment of ejected Vibrio fischeri with ones that have never been inside a squid, see if there's any horizontal gene transfer or sharing of information, so that the previously undigested or unentered squid ones adapt, so they're able to do that without ever having been inside a squid.
Yeah, there is actually, and actually, there's so many generations that happen later that you're talking about many descendants of that microbe that got ejected from the light organ, for that microbe's descendants then to colonize the next generation, because the Vibrio fischeri divides every 30 minutes or so in an optimal situation. And so it's many generations later that microbe eventually would colonize the next baby squid. So there's definitely a benefit to keeping this certain genes. And like I said, this selection pressure gets maintained in the environment in order to keep this kind of association going.
Well, not just descendants, but, all right, let me back up. Have you characterized Vibrio fischeri that have never been inside a squid? Are there ones out there that you've been able to take and compare them to ones that have been spit out by a squid that were successfully living inside one and seeing what the differences are?
Yeah, people have started to do that. They've actually sequenced the genome of Vibrio fischeri that just never colonized squid. And then there's some that they've sequenced the genome to look for what are the characteristics that these Vibrio fischeri need in order to colonize. And there are differences. Like if you took a Vibrio fischeri from the Atlantic Ocean that has never seen a squid, and then you sequence it and compare it to a Vibrio fischeri that just got ejected from a squid ventate, there's a lot of differences. So the genome of Vibrio fischeri, you're right, it's malleable. It can exchange genetic information, as do most bacteria. There's constantly swapping of genes and plasmids, but if they swap out something bad and that doesn't allow them to colonize the squid, then they kind of get removed from that population. They kind of lose out. So there is even though there's a lot of genetic swapping of material, there's always that constant background of selection pressure for us to become what we call a symbiosis competent bacterium.
But if you put together naive and I don't know what you call the opposite of naive.
Oh yeah, if you do a competition, like if you put two guys together and tried to compete them, you'll see differences. Only the guys that know how to colonize get in. You can definitely do all kinds of what we call competition experiments, where you might add a gene or take away a gene and ask, okay, how did this affect its what we call fitness in its ability to colonize and thrive? Not only, some microbes.
I'm not saying.
Okay, go ahead.
I'm not saying competition. Like I said, have you taken naive Vibrio fischeri and had them hang out in the local environment with experienced ones and see if the experienced ones inform the naive ones, so that without ever having been inside a squid, they're able to do so successfully the first time, more often than just naive ones that have never been informed?
Well, if it's of the right strain, if it's the right strain, then a naive Vibrio fischeri can colonize no problem. But if it's a different strain, then no, it would have to undergo some sort of genetic manipulation in order to enable it to colonize. So, but we have naive Vibrio fischeri all the time that we colonize the squid with. So almost every single experiment I do, that Vibrio fischeri has never seen the inside of a squid. So it's really what we're just taking. We're taking a strain of Vibrio fischeri that has been, it's passed down its essential genes, and so that's pretty much every experiment we do, and that these Vibrio fischeri have never seen the inside of a squid.
So are you looking at, you said you know what they eat inside the squid. How about what they excrete or the signaling that they do? Have you analyzed that, and have you found anything interesting there?
Yeah, there's a lot of interesting products, and we don't know the full extent of all the different molecules, but one molecule in particular that seems to be really important for the host, for the bacterium, is a molecule called lipopolysaccharide, and also there's several of them, but I'll just talk about that one. And that molecule seems to trigger the developmental change inside the host. So the host kind of receives that signal, and then it undergoes a developmental remodeling because of that signal. We call it apoptosis, or programmed cell death. And so once the bacteria colonize, they're kind of releasing signals as well. And we don't understand the full cascade and, which, the entire pathway, but there's definitely examples where the bacterium is manipulating the host to release certain goodies for the bacteria to survive, and also it's the bacteria releasing certain signals that diffuse outward and can trigger changes within the host. So it's a really interesting way, again, to study how the effects that the microbes have on the physiology and the response of animals.
Okay. So what are your goals with the research? What are you hoping to figure out?
Well, my little piece of the symbiosis research community is I've actually been trying to apply this to space biology. So we currently have an international space station, and we have plans to go further out and go back to the moon and hopefully to Mars in the next few decades. And in order to take humans out of the biosphere, we really need to understand how microbes are going to be affected by the space environment. Specifically, I look at microgravity. There's other things like radiation and other factors in the space environment, but I'm really interested here. I'm down here at the Kennedy Space Center, and we're very interested in how the space environment is affecting that balance between health and disease, between microbes and their animal hosts. And that's kind of been the work I've been focusing on in the last few years.
Okay. Anything interesting you've seen? Have you gotten experiments onto the space station and evaluated what the changes have been?
Well, I've had experiments up on the space shuttle back in 2011 on the last two space shuttle missions, and I'm scheduled to launch to send another experiment on the International Space Station. But the first good thing is that our study showed that the bacteria, the Vibrio fischeri, was able to find its way into that light organ, and so I use the squid again as a model because it's so much smaller and simpler. But we were able to get positive colonization in the space environment, which was good. We weren't sure how microgravity would if it would disorientate the bacteria or change their behavior. So that was a good thing. The other more profound results that we've been finding doing microgravity related studies is that it seems that when you don't have your microbes. So the good thing about the squid is we can grow them without bacteria. So we can see what happens when they don't have it and when they do have it. And when we grow them without bacteria and put them under the stress of being in microgravity, they actually don't do so well. They actually have problems. They start to show different kinds of stress responses. Their immune system kind of goes haywire. And however, when we add Vibrio fischeri to the animal, the immune response seems to calm down. We start to see less oxidative stress. We haven't tested other stresses, but that's the main one. So it seems like Vibrio fischeri is helping to modulate the stress response of the animal in the microgravity environment. So that could actually have some implications for human health as well. That because one of the things that happens to astronauts as well as squid is that their immune systems can become compromised in space. So we would need to be very careful about what microbes are present, whether it's on their food or on the surfaces of the spacecraft or interacting with their bodies. So we've been trying understand, what happens under this stress environment. And what we found is beneficial microbes are really critical for, at least for squid health, under those microgravity conditions. So we've been publishing those results over the last few years.
Well, how are they important for squid health? What are they specifically doing for the squid?
So when they get into the light organ, one of the things that happens in microgravity that we're seeing in the squid is that there's an increase in the production of what's called reactive oxygen species. These are things like different molecules of oxygen that are really reactive and can cause problems for the host. So what we've been finding is that in the stress of microgravity, there's a lot of these oxygen radicals. And we're finding that when bacteria are present, they're kind of turning down, or they're helping the squid deal with that oxidative stress environment. So having the bacteria around is actually helping them cope. This is all at a molecular level with being in a more oxidatively stressful environment. So that's one angle, and the other thing that they're doing is they're seem to be regulating or modulating the response. So they're helping the squid kind of tone down their immune response. So sometimes when there's no bacteria, they upregulate all these different kinds of immune genes, and that can be stressful for the animal. And so the bacteria kind of calming things down molecularly and helping produce fewer of these molecules associated with the immune system.
Well, how are they doing this, do you think? Literally, by what mechanism?
That I don't actually know. So we're working on trying to replicate this in the lab. And one of the things that we're hypothesizing is so squid have there are these things called macrophages that our bodies have that kind of go out and they sniff around. And if there's a microbe they don't like, they kind of phagocytose that microbe and then they destroy it. Squid have very similar cells. We call them hemocytes. And in the microgravity environment, those hemocytes don't move where they're supposed to go. They kind of just don't migrate very well. And so what we've been trying to figure out is why is microgravity affecting the movement and what we call the trafficking of these immune cells to where they're supposed to go. And we don't know if they're not getting the signal. So it could be that that something about microgravity is not transmitting the signal to the immune system to respond properly, or if the cells themselves are getting the signal, but they're not responding to the same extent they would as if there was gravity around. So we're right now trying to figure out, is it the signal or is it the response that is different in microgravity? And so we've designed a few experiments that my graduate student is working on right now to look at signature, like try to artificially boost the signal to trigger some of these microbes, to trigger the movement of these immune cells. And we're creating what we call a simulated microgravity environment where we're replicating some of the physiological factors associated with microgravity to try to suss out some possible hypotheses that later could be tested on the International Space Station. The problem with doing any kind of experiment in space is it's really expensive. So we try to use these, they're imperfect, that is for sure, but we use this modeled microgravity simulator that will help us try to develop testable hypotheses that we can then test in space. But we're trying to isolate whether it's the signal or the response, and that's kind of where we're at right now. And I don't have more information for you. Hopefully, you will.
Well, I guess the cells and the bacteria are sensing gravity. They must have some component of their cells that can.
They are. It seems like they're changing, and you would think that something so small with the forces that they wouldn't. but they do seem to have different physiological responses in microgravity. And one of the things that we've been seeing is they actually seem to, I talked about this molecule called lipopolysaccharide. It's an endotoxin that a lot of bacteria use to communicate with animal cells. And in microgravity, what's happening is the membrane of these bacteria are getting weaker, and they're actually causing more of this endotoxin to slough off into the environment. And that might be part of the reason why the squid are changing some of their physiological, I didn't really talk about that, but they're changing their behavior as well. And so we're trying to figure out, what is it about not having gravity that is changing, that is weakening the membrane of the bacteria? And we're just trying to figure that out right now. But we definitely are seeing physiological responses. Bacteria tend to grow faster in microgravity. Their membrane is weak. They're definitely changing their behavior in the absence of gravity.
Well, if you look, what's the approximate diameter of a bacterial cell that you're studying?
About 1 micron, so 1/1,000,000th of a meter.
So if you look at, like,
So they're
if I
tiny.
imagine the bacteria is like a sphere sitting on a surface, the top is the gravity differential between the top of the sphere and the bottom of the sphere, when it's on Earth, it's somehow able to sense that because it loses that sense in space. And that would be a
Absolutely.
incredibly tiny differential, I would think, that the bacteria is able to sense across that tiny distance.
Absolutely. And I think that it has to do with shear forces. And so this is pushing my limits of understanding in terms of, I am definitely not a physicist, or fluid dynamics person. But what also is what's happening in microgravity is that you're getting a change in the shear forces that's happening in the liquids. And so the microbes are growing faster, and that might be, especially this particular bacterium is motile, and it does seem to respond differently to these lower shear forces than it would if there's gravity pushing down them and maybe causing a higher shear force. So it could be, even in these, like you said, these small scales, we do see physical changes in what's happening in their microenvironment.
Have you tested uh, how sensitive they are to chemical gradients? Have you looked at adding just literally just a few molecules of something to solution and seeing if they could sense the chemical gradients on those?
They definitely have chemotactic responses. They definitely have that. And in the case, for example, sugars, you can actually create, they love chitin. This particular microbe loves chitin. It's a sugar. And chitobio, excuse me, that's the sugar. And you can set up a trail and create chemical gradients to lure them in. And that's actually how the squid attracts some of the Vibrio fischeri in, is it sets up this chemical gradient, and the squid swims up there. And that's why we think that there's not a problem in space of actual colonization, because the bacteria are probably following this chemical gradient. So yeah, that's a good observation you made.
Also, too, I guess a pressure gradient would be developed if it wasn't in space, but if it is in space, it might not. There's the weight of the fluid above a certain point that would create pressure on it. And if there's no weight to it, because it's weightless, then there'd be no pressure.
Yeah, I'm not sure about the changes in the pressure. So I'm not changes. So in the habitat that we've tested, there should not really be changes in pressure. We've tried really hard to it's an enclosed space, and in terms of partial pressure of the environment, it's the same pressure as if, the partial pressure of the atmosphere, so to speak, is the same as if it would be on Earth. It's still 14.1 part PSI or something like that. So that shouldn't change in the little hardware, the flight hardware that we're actually using. But you're right, I haven't really looked at that. So there could be issues there with the actual physics of that environment, especially the fluid dynamics. I haven't explored that in the hardware yet that we're going to be using.
Okay. Well, very good. I guess last question. Have you looked at what these bacteria, again, are excreting, and seeing if that's a food source for the squid, or what they are excreting and what happens to those chemicals?
Yeah, a lot of those chemicals kind of diffuse across the membrane, and a lot of times we don't have a full understanding of what the squid is a complete composite of all the molecules that the squid is passing through. But there does seem to be a protein there does seem to be several examples of that the microbes are kind of triggering the release of little vesicles by the cells, the host cells lining, and the bacterium are kind of activating to release. And we don't know yet if that is a physical, like a cell-to-cell contact, or if this is a diffusible signal. But the presence of the Vibrio fischeri will actually release little vesicles to be released, and it's just a huge amount of food for the bacterium, and then they can eat them. So we're still working on all the different dynamics of the molecules that go back and forth between the host and the squid. But there definitely seems to be a lot of activity by the bacteria in order to manipulate the cells around their little habitat, their little crypt space there, in order for them to activate the squid in order to feed them. So it's kind of like maybe a baby bird triggering the mom's reflex there to provide food for the bacteria.
Okay, very good. Well, so what's your what would be like a happy result for you in the next couple of years? What kind of results would you be thrilled to see?
Well, first I'd like to see if we can replicate some of the experiments we've been doing on the ground in space, and really see if the bacteria truly are modulating and controlling the immune system. And I think then that information could potentially translate to more human cell models. It's very hard to do experiments on actual humans in space. For some reason, astronauts don't like to be dissected. I don't get that one, but that was sarcasm. But anyway, we are trying, hopefully the end result would be some information that will help mitigate potential health problems for the astronauts, and that they can use that information to deal with potential microbiome related diseases that astronauts might be susceptible to as they're traveling beyond Earth's biosphere.
Okay. Well, very good. So what's the best way for people to get in touch, to ask questions, to see some of your papers, to, adopt a squid?
Well, I have
Just kidding.
Adopt a Squid. We actually had an Adopt a Squid. A couple of my students started that. Yeah, it was on experiment.com. They were actually doing a specific experiment that they were doing, a kind of a GoFundMe site for scientists. And they've been publishing that result. So that was possible. But my website, jamiefosterscience.com, is probably where you can get a lot of information, some papers, and that has my contact information. Again, I'm with the University of Florida, so if you just Google me, Jamie Foster at University of Florida, my website comes up, and people are more than welcome to contact me, email me if they have specific questions.
Well, that's great. Well, Jamie, thank you for coming. I really appreciate it.
Yeah, no problem. And thank you for inviting me.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.